I'd like to invite Annalie Melin, who is going to be talking on new taxonomic insights into long-legged oil bees, exploring the Great Escarpment of South Africa.
Good evening from Cape Town in South Africa. My name is Annalie Melin, and I'm a bee researcher based at the South African National Biodiversity Institute. I'm going to talk about some taxonomic work I have recently done on oil collecting Rediviva bees. So approximately 3% of the world's bees collect oil from flowers. This has led to specialized morphological adaptations for accessing floral oils. You can see the floral oil glands here in the base of the flower, the bees reaching in. They've developed blade-like hairs to be able to burst the oil glands inside the flowers, and they have specialized hairs for mopping up the oils. Rediviva is an endemic bee genus in South Africa and Lesotho. It's a striking example of pollinator adaptation, particularly their elongated forelegs, which tend to match the spurs of their host plants, which is possibly driving their diversification. So although this research focuses on taxonomy, the project began while I was studying trait evolution of Rediviva bees. I found some intriguing variation in the species Rediviva nelliana. Unlike other Rediviva species, its allometric relationship between body size and foreleg length showed that it was an outlier. So you can see the graph here on the left with foreleg length on the y-axis and body size on the x. And you can see that the Rediviva nelliana is here in yellow, and its slope is much steeper than you see for all the other species. I also found that there was an unusually high variation in foreleg length and a wide species distribution. You can see the pink species distribution model here. And the emergence times tended to vary depending on location. This all suggested that it is a group of species which we call the Rediviva nelliana group. So delving into the taxonomy, in 1931, Theodore Cockrell described Rediviva nelliana based on a single male specimen. And then 3 years later, he described Rediviva polytissima based on a specimen found some 300 kilometers away. And then over 50 years later, Van Whitehead and Kim Steiner synonymized these two species as Rediviva nelliana, which was later redescribed by Whitehead and colleagues. Now, the revision didn't include sufficient detail or illustrations of the diagnostic characters of the male, and as such, the justification for synonymizing the two species are unclear. The taxonomic problems around Rediviva nelliana were further complicated by the description of two new species, Rediviva steineri and Rediviva whiteheadii. My examination of an extensive collection of Rediviva nelliana revealed that these two species were included in that collection and therefore form part of the Rediviva nelliana group. So the next step was to look at the key morphological characters used to define Rediviva nelliana. In both the males and the females, the main diagnostic character was the shape of the mouthparts, and specifically acutely pointed gallia. So I examined a large amount of old material, about over 1,200 specimens, and the type specimens. I also surveyed different sites across the Great Escarpment of South Africa, and this included revisiting the type localities. Through this, I found clear evidence and differences in the shape of the galea, which is consistent between the sexes and in the head and sterna in the males. So rather than being acutely pointed for Rediviva nelliana, you can see that it is rounded and more beak-like, and this is consistent between the male and female, whereas Rediviva polytissima is acutely pointed and falcate. I found that Rediviva nelliana, Rediviva steineri is rounded apically, whereas Rediviva vogeli is spear-shaped and pointed. So in addition to the shape of the mouthparts, the male's head and sterna also proved to be diagnostic. In particular, the ventral margins of sternum 8. If you have a closer look, you can see how they vary. And one key discovery came during my field work when I collected a mating pair on Wallenbergia flowers. And this allowed me to finally identify the previously unknown females of Rediviva nelliana. But this also revealed that the females identified as Rediviva whiteheadii were in fact Rediviva nelliana. So in conclusion, the Rediviva nelliana group is comprised of 4 species. And I redescribed Rediviva nelliana and reinstated Rediviva polytissima as a valid species. The male of Rediviva whiteheadii has been assigned to another species, Rediviva autumnalis, and its female was synonymized with Rediviva nelliana. I also reassigned over 240 specimens from Rediviva nelliana to Rediviva steineri, and which vastly expanded its known distribution. Additionally, I described a new species, Rediviva vogeli, and this was named in honor of the late Professor Stefan Purkel, who described the pollination relationship between oil bees and their host plants. And finally, I updated the key for 12 Rediviva species that occur in the summer rainfall areas of South Africa using the mouthparts and the male head and sterna. So in light of these results, I plan to reevaluate the relationships for Rediviva nelliana. In the past, I treated the data as populations, but now know that it is multiple species. And similarly, a study in 1990 assessed the size matching between Rediviva nelliana and its host plants. And again, it was also treated as a single taxon, but now we know it's 4 species, so this relationship would also need to be reanalyzed. And finally, one would need to redo the phylogeny for Rediviva to assess the forward four-leg evolution. Resolving the taxonomy of these specialized bees not only aids evolutionary studies, but also uncovers hidden diversity. These research outcomes will contribute to conservation efforts such as red listing. And prior to this work, Rediviva steineri was only thought to occur at 3 localities, and you can see those highlighted in turquoise there. And as you can see, the species distribution shown in either red dots or a species distribution model, that the distribution for the species is significantly larger, which will produce a different threat status for the species. And I think that is all. Thank you. Thanks for listening.